Good morning, everybody. This is the 29th of our online seminar series in Biodiversity Informatics. I'm Town Peterson, that's Kate Ingenloff. This series has been going for 3 years now. This is the 29th in the series. Today we have Steve Kelling from Cornell Laboratory of Ornithology with us. I'll introduce him in just a second. I want to remind you first to send questions.
We lost you.
Did you lose me? Can you hear me?
Yeah, you're muffled.
Ha! Can you hear me?
Yeah.
Okay.
You can move back now.
Okay. So you can send questions to this email and this Twitter account, either one. We'll see them. I would urge you to send the questions as they come to your mind rather than at the end, so that we don't have a bit of a delay between the end of the seminar and the arrival of the questions. So there are those, biodivtraining@gmail and my Twitter account. So Steve is going to give us a talk on essentially citizen science in the biodiversity world. He's going to give us a perspective from the Cornell Lab, which has been one of the major, if not the dominant, mover and shaker in this world of citizen science linked to science applications. So I'm quite excited to see what Steve has to say. And with that, I'm just going to pass this off to Steve. And Steve, I'm here, and I'll let you know if you blink off.
Okay. Thanks, Town. Good morning. I'm just going to start by turning my PowerPoint on. So you'll need to tell me if you see my PowerPoint.
I'll let you know.
Doesn't sound like you see it. So.
No, I don't see it yet.
Okay. Remind me how to do that.
Left side of your Hangout screen, the green rectangle.
Okay.
Looking good. Hold on. Okay, now I can see your wallpaper and your Hangout screen. There you go. You're on.
Okay. Great. Thanks. So what I'd like to talk about is how we use citizen science to monitor biodiversity. And the basis of this talk really came from a meeting that I was at last March, which was looking at the challenges of monitoring biodiversity globally and what actions can be taken. And I was part of a group that focused on citizen science. And what came out of this is I included some more people from the Lab of Ornithology on this. And what I'm going to talk about is a paper that we've recently submitted to discuss this challenge and look more broadly at all of the different kinds of citizen science that's going on that's monitoring biodiversity. So we're experiencing and the other thing is this is the first time I've given this presentation. There'll be parts of it that I'm going to read, parts of it that'll be sort of rough. I apologize for that, but it's hot off the press too, so you get the good and the bad. So the concept is that we're really experiencing losses in biodiversity with the most threatened species or regions having the least amount of information. While local and species level data are essential, the number of biodiversity assessments that actually functioning are relatively low. And over the past couple of years, there's been numerous authors who've suggested that recruiting the public in citizen science could provide large quantities of species occurrence data over broad spatial and temporal extents. Now, there's hundreds of citizen science projects that are already enlisting the public to collect species observations. Most of these have very limited numbers of participants, small geographic scope, and often are short duration. However, we all know of, particularly in the US, several long-term monitoring programs like the Christmas bird count, or in Europe, the butterfly monitoring programs. And these projects are generating millions of observations annually from really pretty large volunteer networks. But while citizen science can collect large quantities of data, many projects most projects are motivated by multiple objectives that are potentially incompatible. For example, in order to engage large numbers of participants, projects have to be fun and educational. But really, in trying to achieve solid scientific objectives, they typically require that data are collected following specific and often rigorous protocols. And this limits volunteer motivation. So a real significant challenge for citizen science efforts is to achieve those sound scientific objectives while still finding ways in which they can maintain numbers of participants. And believe me, this is really easier to say than to accomplish. One of the key issues with citizen science project, as well as all monitoring projects, is really attempting to control for data collection biases, which are inherent in any kind of monitoring project. So some of the data collection biases, for example, that we experience is uneven data collection over space. And this is a map of Missouri, and this is from 4 or 5 years ago. And these are locations where people have submitted observations to the Lab of Ornithology. You can see the major thoroughfares. You can see the large cities, St. Louis, things like that. And then there are areas that are blank with no observations at all. The other thing is there are uneven skills in which participants can detect and then classify species. And this is a series of graphs out of a paper we submitted last year, which shows that we use the concept of species accumulation curves, something that's very long and tried and true methodology in ecology to understand the biodiversity of a location. So in this case, we fixed the location and then looked at different individuals and measured their accumulation rate. And what we found is that, which I'm sure you would agree with, is that some observers are good at accumulating species quickly. So that's on the right side of the screen. They have a high species accumulation rate over time, where others have slower accumulation rate. So we have to account for these differences. And then the final is that there's uneven detectability of species. Birds sing in the spring and they're rather cryptic in the fall and winter. And this graph shows that for species that vocalize, they're often easier to detect if you know their vocalizations, where those that aren't are less so, or different times a year are less so. So there are various data collection methodologies that vary from very unstructured and open data collection processes, which really don't try to control for any of these data collection biases, to highly structured protocols that do control for these biases. What I'll do now is compare and contrast the differences between these unstructured, sort of mapping the unknown projects versus these more structured monitoring biodiversity projects. And I'll note the significance of broad participation in the unstructured projects. So there are lots of people participating, but the data quality is not that great, versus the more structured projects, which is really hard to keep people participating in. So again, unstructured projects tend to be open. Anyone can participate. There's very little information provided on how to collect the observation. Most of these observations are opportunistic, but they have broad participation across broad spatial extents, but the data quality is pretty low. You usually get presence-only information. More structured projects are motivated by a protocol, a question, and the question controls what is collected in the protocol, and the protocols then try to limit these biases. The data quality in these are really high. The inference you can get out of them are very high. But participation and long-term participation is difficult to maintain, and often the costs of these projects are prohibitive. So we started with a hypothesis that by applying basic improvements to sort of these unstructured data collection methodologies, we can greatly increase the quantity and quality of the information that can be obtained in citizen science projects. So we've really come up with a middle ground, which we call semi-structured projects, where we use protocols that collect sufficient effort information to allow analysts to manage these biases. But at the same time, we allow open participation and are open for participants to submit observations from anywhere. In thinking about these biases, I did a lot of exploration in the literature, much more broadly than just in biodiversity monitoring, and one of the things that I kept running into is this little poem from Rudyard Kipling's The Elephant's Child, Just So stories. These were written about 100 years ago. You know, they're very untrue information about how does an elephant get its trunk. But the important component of this is I keep 6 honest serving men. They taught me all I knew. Their names are what, where, and when, and why, and how, and who. And this has been termed the Kipling checklist. And the Kipling checklist is a list of standard questions that are asked during what's called root cause analysis. And root cause analyses is the process used to investigate and categorize the reason an event occurred. This methodology is often used in industrial safety and health scenarios. And the important factor is that the root cause analysis not only identifies what event occurred, but also why it happened. So, for example, an observation would be, or the symptom would be, the appearance of a weed. But the underlying cause of why that weed is there, how that weed was observed, and all of those features aren't obvious from just the observation that there is a weed. So when we apply this notion, this root cause analysis, to citizen science, the Kipling checklist really provides an objective foundation for the clear planning of a survey designed to document the occurrence by answering these why, how, what, where, when, and who questions. And the quality and the way you get answers for them will determine the downstream utility and applicability of the data collected in these projects. So let's go through them quickly and get a sense. So why is the survey conducted? That to answer the why conditions, really how to answer the rest of the Kipling questions, and is the foundation of nearly all the decisions about the project design. A disconnect between the why and the protocol design will limit project success. So, for example, if all you're doing is collecting positive observations of the occurrence of the species, but you're interested in looking at trends over time, there's a disconnect between the positive occurrence that you're collecting and the ability to measure those trends. Data collected about how much effort, for example, the duration of the collecting event, the distance traveled while collecting, really provides crucial information needed to estimate abundance of species, detectability, or completeness of effort. So the more information you collect on how observations were collected, improves the inference from the data you collect. We've found that surveys that target species within a specific taxonomic group and require participants to report all detections and classifications of those species provide critical information that allows analysts here to know what species were detected, as well as to infer species absence, which we call non-detections. And when surveys are repeated at that same location, we can estimate detection rates. Knowledge of the locations of where the observations allows connections to be made with features of the environment. So environmental factors not only constrain where species can live, but factors such as the structural complexity of the habitat, temperature, or other climatic conditions can influence the activity patterns of the organisms, ultimately influencing their detectability. And then species detectability can change daily or seasonally as behavioral characteristics of the organism changes. For example, birds vocalize more frequently in the morning in the spring and during the breeding season, and are more easily detected during those periods of time than other times of the year. Finally, while volunteers can develop enormous expertise in gathering information on species occurrence, observers exhibit tremendous variation in the detection and classification skills. and the resulting quality of their data. As such, then, some training and or information on the level of expertise can be helpful to data validation purposes. So now I've taken all of the Kipling checklists on the left column, the why, how, what, where, when, who, looked at the data collection methodology and what the outcomes are for structured, semi-structured, and unstructured survey designs. So for unstructured, the real concept is really determining where species occur. For so much biodiversity, we have no idea where species are. And to best collect that information, how people collect these observations is relatively open, often opportunistic. What they collect is often open and opportunistic. Where they can do that anywhere, when, anytime, and the participation is open to the public. For structured projects, how the data are collected is more developed and specified by the organization collecting the data. What's collected has been predefined. The organizer identifies where and when, and often the participants are recruited and trained to collect these data. Now, the semi-structured fits somewhere in between. The why can be for mapping the unknown, or the why can be for monitoring biodiversity. The how is recorded, so the observer can choose how they collect the data, but information on the amount of time, the distance traveled, and things like that are collected. So what's collected has been predefined, but people can collect anywhere, do it anytime, and participation is open. So now going down to the bottom and thinking about the outcomes of this, unstructured projects give you estimates of presence only. Semi-structured give you information about estimates of detection probabilities. They give you information on the presence and then non-detection by the participants. They can be used to estimate abundance of an organism. And I'll show you some preliminary information with some of our semi-structured data, but structured data allows you to get to trends and to be able to see how species patterns change over time. So now I'll quickly review and give you some examples of each of these kinds of projects. So the concept, as I've been saying, for unstructured data is mapping the unknown. We need to know where things occur before we can begin to think about conserving them. These projects can recruit lots of volunteers. And what the kinds of results you get are exploratory or visual. You can create maps of occurrences of species and things like that. And a good example of this is from the Map of Life project. Map of Life goes out and they look for positive observations of a species over broad spatial extent. So each of these gray dots are locations where some collection has information on the occurrence of a wood thrush. Then they use an expert opinion to sort of draw a map around these dots. And you can see there's some interesting features of the map, like this area going up through South Dakota where there's no observations, but the expert who drew the map had evidently had some indication that they occurred there. And then some of the observations aren't part or fall within the design of the map. The gold in this case is the expert's opinion of its breeding grounds. The tan is their migration, and blue is where they winter. So the points are removed and you get a distribution map that looks something like this. So these kinds of maps are actually pretty good at broad scale and coarse grain, but as you drill into them, as you try to get at higher levels, they begin to fall apart. Probably the best example of these kinds of data collections now is iNaturalist. iNaturalist goal is to document the presence of all species anywhere on the planet. And participants are primarily amateur observers. They use an easy-to-use mobile app and web tools to document basically opportunistic observations. So the workflow is you go out and you see an insect, you take a photo of it, submit it to iNaturalist, and a group of experts review what the insect was and identify it. And if multiple experts identify it similarly, that's considered a research grade observation. And that research grade observation with the image then is provided to GBIF, the Global Biodiversity Information Facility. Now, more structured projects really are fundamentally based on the notion that informed conservation most efficiently is conducted when it's based on a priori goals. The why of the Kipling checklist is very well defined. And those goals can direct the survey design to allow the data collection to be specific for the kinds of analyses you want to do and the answers you want to get. So it's a very linear workflow. You develop the question, you create a study design, the why, you collect the information that you want to collect, analyze the data, and you get an answer. These kinds of projects really generate the strongest degree of inference. When structured surveys are repeated, they provide accurate information on the status and trends of species over time. This is a really excellent example from the North American Breeding Bird Survey. And this is a map showing 50-year trends of the wood thrush throughout the United States. And the red areas are showing negative trends of about 1.5% annual decline. The blues are 1.5% positive declines. This is, in my opinion, the state of the art. These are really excellent analyses of well-designed data. But there are other kinds of projects doing this too. And most of these are atlases. Because it's so difficult to run these projects and recruit participants. For example, for the Breeding Bird Survey in New York State, only half of all of the routes are run. Typically then what countries will do is have atlas periods where they'll really recruit and invest a lot of money to get participation periodically. And atlases are typically run for about 5 years. And very specific survey designs are used. And you can develop these very accurate abundance estimates of species in the atlas period, and then look at how those abundances change across times of the atlas. So what we're focusing on now is the compromise. We want to be able to collect data that we can get as much inference out of as possible. Our goals are really to try to look at the status and trends of populations over time. But we want to have open participation. We want to collect lots of data. So we need to incentivize participation in the projects. And our concept of our study design is that we have a data collection process that's in place, constantly running. I'll go back to this at the end. And we provide data exploration, so maps and visualizations of the data to allow anybody to begin asking questions about the data. So that's part of the unstructured component. Where we change, though, is that we allow posterior hypotheses to be developed, where we can take subset out either parts of the data or actually modify and add protocols to the data to collect specific kinds of information, then do the data analysis and get the answer. And the project that I'll focus on to describe that is eBird. eBird is this notion that we've come up with at the lab about 22 years ago, where we thought that we could find some value in the birders' checklists, and if we could organize birders to submit checklists in a fashion we might be able to use them for research and conservation on birds. So we got a grant from the National Science Foundation in 2000. We started the project in 2002, and for the first 3 years of the project, we got very little participation. So we were thinking, what's going on? What did we do wrong? Why is this not working? And we started asking people, and their response back to us was, you got to understand your community. And in this case, our community is not only the stodgy old birders or ornithologists who really know patterns and are very biased or questioning the ability of the masses, and typically in our case, the young masses of birders out there who are submitting observations in this formal way, as checklists and as part of their hobby. So what we did was we changed the whole concept theme of eBird from what can birders do for science to what can we give birders the functions that they want to get them to participate. And one of the big things that birders love is managing lists. And any of you out there who are birders know you have probably year lists and life lists and day lists, and all of these lists are based on taxonomy. And when the taxonomy changes, your whole list will change. So if we could develop a way of managing automatically participants' taxonomies for all their lists, that would be a feature birders would like. We also decided to focus on developing web apps and mobile apps to allow data collection to be easier. And then we provided all kinds of features that allow people to explore the data, either through maps or prepare trips by looking at regions. But one of the things that we've found that birders like, or participants in crowdsourcing and citizen science like, is their names on lists. So here, for example, is my yard list. This is what I'm mostly interested in. My yard list is about a little over 200 species, and I rank 63rd out of all the participants. Although I submitted a lot of checklists, so I'm ranked 5th in the number of checklists submitted. But here are some yard lists from all over the world. And you get your name and what species you've seen and things like that. People love this stuff. And then recently we've added a sort of a poor version of a Facebook page based on the eBird data for anybody who wants to join in. And you can write a little biography of yourself. And this pulls out information about all the states in India where Early has made observations. And we now collect photographs too, so those photos get posted and things like that. All of these kinds of things have really gotten the birding community, the people we wanted to most engage, really excited about the project. And since 2006, the growth in eBird's been exponential. We've collected now about just under 400 million observations. It took us 10 years to collect the first 100 million observations. And in 2017, we will probably go over collecting 100 million observations. Last May, for example, we collected more observations, about 12 million, than we did during the first 8 years of the project. So we're growing. We think that we're providing services to the community to keep them motivated. In 2010, through pressure from our community, we opened up eBird to go global. And after 2010, this is not a map of the world. This is a simple plot of all the locations where we had eBird observations. This is 2016. So you can, these are interesting plots. They show you where we have data deficits, for example, in central Brazil, so we have really focused building communities there, North Africa, Russia. But we're really proud about our growth and participation in India, South Africa, all through Southeast Asia, Australia, and a lot of South America. And we're having an impact. This is from a paper from a group at Cambridge that's showing the proportion of eBird records in GBIF, and the contribution that eBird has provided to GBIF in general. And all the red locations are countries where eBird data, it represents about more than 90% of all the data, biodiversity data collected. So what can we do with this data? We can visualize it in these really exciting methodologies that allows you to look at a population of birds through its entire life history. So you can see where wood thrushes winter in the Yucatan, move quickly up in the spring into their breeding area, then much more slowly back down to their breeding area. These kinds of data visualizations allow people to begin asking questions about why is their migration more fast in the spring than in the fall? What are the drivers of that? How much time do they spend in different regions? And we're collecting this data now throughout the entire Western Hemisphere and coming up with observations and these kinds of animations. In this case, for an austral migrant, the fork-tailed flycatcher. This is hot off the press. This bird winters in the equator of South America. There's some that are up into Central America and the Caribbean, but then during their summer, our winter, they move down into Argentina and Uruguay and then back up. So these kinds of visualizations really are great in allowing people to begin asking questions and allowing people to begin to see how an entire population of a species moves through the environment. We've done this, made these kinds of visualizations for 400 or 500 different species now. So really what we have to do is think about what the processes of science is, particularly when we're looking at bird populations. And new ideas really come from the exploration and visualizations of patterns within the data. They're seldom come from preconceived ideas, some light bulbs going off and some testing of an experiment is very rarely done. It's more looking at patterns and exploring, visualizing and thinking of questions. And then from those explorations, researchers can then properly conceive of the questions and develop confirmatory analyses to identify the causes of how species occur in the environment. So we're thinking that our belief is that this semi-structured approach, where we combine the data collection processes with the exploration, being able to view the data, ask questions of the data, and then have a hypothesis driven either from the existing data or subsets of the data, or creating new protocols that we can put into the data collection methodology, really provides an opportunity, a big data approach to being able to look at populations of birds over space and time. And some of those questions and hypotheses that we've been playing around with at the lab, one was the question, could we use eBird data to look at trends of bird populations across their entire life history? And this map on the right is our first example. We've done about 40 species where this is, again, the wood thrush. This is an 11-year trend from eBird data showing roughly in the same general area that BBS is showing, about the same amount of decline in that species, about 1.5% per year decline. But what's interesting is in the areas where they winter, because of that concentration of the species, you can see the decline really more generally in the population is greater, almost 2%. We also can look at questions about, well, where do these neotropical migrants spend most of their time? And this is a map of a paper that's just about to come out in Proceedings of the Royal Academy. that shows that the number of days, we call this a use days map, of approximately 20 species of neotropical migrants. And what you see is this area in the Yucatan and through Central America for these 20 species, they spend more than half, upwards of 60%, some as much as 80% of their entire life history in these small areas in the Central America, and then radiate out and spend during the breeding season a significant amount of time, but less so over much broader spatial extents. This is allowing us to be able to see what the potential bottlenecks are in the conservation of bird populations. So in summary, really the goal for a citizen science project is to identify really what kind of information you want to collect, the why, and then develop a method of surveying that provides you the opportunity to really collect the meaningful information to answer that why question. I can't say enough for the significance of providing incentives for participation. There's a broad literature in the crowdsourcing concepts, human computing interactions, things like that, that emphasize this same thing. We've really focused on building mobile apps to collect data. The mobile apps we're evolving to now, you press start and stop, and it keeps track of all the effort information, and all you do is identify the species and give counts of them. Let the participant do what they really enjoy doing, which is identifying things, and use the app to collect all the effort. And this semi-structured approach allows us to be much more broad in the kinds of things that we can do. So we can map the unknown with eBird data. We're getting information, for example, in India about where species occur, which there's very little information on. But at the same time, we can incentivize participation by volunteers to collect more effort-based, more higher quality data that allows us to do more with the information we're collecting. So with that, I'd like to thank you. We have lots of people, foundations that we really we always have to acknowledge, but for us, it's our partners and volunteers that really motivate the science that we can do. So thanks. I don't know what I do now, Town. Do I just say stop?
No. You go up to the green rectangle and tell it to stop sharing your screen.
Stop sharing this. Okay.
Coming out of it. Now go to screen share, the green rectangle at the left.
Yeah. Don't share it anymore.
Yeah, stop sharing.
Okay.
And we should see your beautiful face. There we go.
There I am.
Okay, thank you, Steve. That was a neat overview. We have time for questions. That's a comment to the people watching. I'll put up the email address and the Twitter feed. There we go. Oh, there we were. There we go. Do send a question quickly if you have a question to either of those places. Steve, just a kind of a curiosity question. What do you do with being simply overwhelming quantities from some places, and then just a trickle from other places? New York City versus the Sahara Desert. I mean,
yeah, we don't we haven't dealt with that challenge, but we have dealt with a challenge between, say, Ithaca, New York, and the county next door, or trying to map a species distribution over broad spatial extents, where some areas you have lots of data and some you don't. And our modeling approach, we call the spatial temporal ensemble models, STEM, is a combination of machine learning, back decision trees, and statistical processing that basically allows us to generate niche models, weekly niche models across the entire range of these species. So those maps that you saw are just the visualization of an enormous amount of data that are generated from these STEM models. And the STEM models effectively make predictions, weekly predictions of the abundance, the relative abundance of a species across its entire range at 3-kilometer interval predictions. So, for example, the wood thrush animation, there are about 4 million predictions in this 3-kilometer grid, weekly predictions across the entire range. So the models generate a surface prediction that allows us to, and then because we collect information on how much effort, we can standardize for that. Because we have information on the quality of the observer, we can incorporate that into the models. And the accuracy of these models, when ground truth or through expert opinion, is actually pretty good.
A question for you from here in my lab group, from Kate Ingenloff.
So first off, thank you very much for your lecture. Lots of great information, and I like the idea of a semi-stratified approach to this. My concern with a lot of the citizen science data, especially when you have a large number of people participating, is that you don't necessarily always have quality control and identification of species, as in you don't know the level or ability of all your participants to identify species accurately. Do you have a recommendation of how different groups handle that disparity in some of the data quality? Are there built-in structures into all of these systems that should be considered that's one is better than the other? And what are your thoughts on that?
Yeah, we data quality is a huge issue, and we and others deal with it in multiple ways. Both say, let's take for example iNaturalist. Most of the observers in iNaturalist are not very good observers. They're often classrooms of participants, but they take a photograph of an object, and they have a series of reviewers, the naturalists, this network of naturalists who actually review the observations, and if they can identify them, and multiple people identify them, multiple naturalists, you gain expertise. The record is accepted. eBird does very much the same thing. We have currently about 2,000 volunteers globally who set a series of filters and review observations as they come in to eBird. And typically they review about 5% of all the data that come in. But at the same time, because I didn't get really into the informatics of all this, but we have a very, very sophisticated database where we can track an observer over time. We can see that observers actually improve regardless of your skill level. Any observer actually improves their eBird submissions the more they submit. And we've also found that we can really measure differences in observer ability and use that as one of the covariates in our species distribution models. And then the final thing that we found is that 90% of the data that we collect are actually submitted by the top 10% of the eBird participants.
Interesting.
Data, you're never going to know what species has been misidentified in any kind of monitoring project, regardless of how good the observer is. You can begin to get a handle on what species were probably there but not detected. So the opposite of that, through occupancy models and things like that, which we can do with the eBird data.
Okay. Well, Steve, you've gotten a couple congratulatory comments, but no further questions.
Okay.
So thanks very much for your time this morning, and thanks for the great seminar. To the broader audience of this seminar series, a month from today, so on the 1st of March, we have coming up Vijay Barve talking about another dimension of citizen science, which is harvesting primary biodiversity records from pictures put up on social media sites. So I encourage you to join us for that. And again, thank you very much, Steve, for your time.
Okay. So long.
Thanks again.